Hello everyone. Good evening and welcome to the week 4's live session. So sorry for the slight delay because there was some technical glitch, so we could not start on time. Yeah. So now in this week 4, you have seen through the lecture that we have gone through different applications of microarrays in the different research, like in malaria research or in the glioma research, along with the different printing technologies. So, of which we will be looking at some other aspects and try to refresh ourselves a bit. So, first thing is that application of the microarrays in the clinical research. So, this is from the paper of the review, which has shown regarding the different arrays and how one can use that for different applications. So like the protein arrays and then finding. So this is an overall workflow that you can speak of or say of for understanding and identifying different markers. There is a NAPA array, and also we have discussed a lot during the uh, previous sessions and previous weeks for the M-NAPA array or the multi NAPA arrays. So M-NAPA or the multiplex, that NAPA is that where the proteins are multiplexed and that spotted accordingly. So that what it helps you to get is that when the proteins are multiplexed, so you will run the samples, and if your protein is belonging for that marker, then you will get that result, and then by resolving one, each 5 proteins or the 5 spots, each spots having different protein sets, you would be able to get the following result. So this was the principle of NAPA, and NAPA is one thing which is of the self-realized. So because the self-based expression systems contains a lot of difficulties and the disadvantages, so that's why it becomes sometimes important to have the MNAPA, uh, to have the self-free expression like NAPA to be there. Next is the Huprot array. So Huprot or the human proteome arrays, where it contains a lot of proteins, all that human proteins printed, and through which serum samples and getting the labeled secondary antibody one can get. Then is the planar antigen arrays or the serum samples, where secondary antibody can be done, and discovery phase and the validation phase with the different kinds of arrays. And the antibody arrays are something where antibodies are printed of the normal proteins and autoantigens are detected. So in antibody arrays, and obviously I think you all are familiar with reverse phase protein arrays. So here in this plate, so here antibodies, so this is the antibodies. So these are your antibodies that are printed. So now we will say that these antibodies are of mutated protein. So you will ask the question that these antibodies are of mutated protein, question mark, protein with having different diseased state. Different disease state. Is it so? It is not. So these antibodies are of normal proteins. Normal means of a healthy living person. So it is of a normal proteins. So now you can ask that what can be the use of such antibodies from the normal proteins. So in a disease state, so in a disease state, what is happening? So in a disease state, whether there are any autoantigens, so detection of autoantigens. So how it is done? If you have antibodies that gets printed of normal proteins, now in actual scenario, no normal protein should react against it, because then what we are saying that this is proteins are not immunogenic in nature, or the antibodies should not be generated. But if there are any uh, auto antigens that are there, so any kind of auto antigens can go and detect and can bind to it, and we can get the auto antigen profiling. So since we have uh, got till here, I would like to give you one uh, hands-on, or give you one resource where you can find this kind of application. So this is for you to appreciate that at how many aspects microarrays can be helping us in understanding the disease biology. So this is the human autoantigen atlas for the autoantigens. So there have been autoantigen atlas also. So with the applications of microarrays and having the different technologies, one can get to know about different articles, different resources for understanding a particular disease can be generated and can be used in different verses and different ways. Okay, so this is auto antigen atlas. So here I'll just give you a brief for you to understand in a better way. So the auto antibodies refer to the antibodies that target your self antigens, right? which can play pivotal roles in maintaining homeostasis, distinguishing normal from tumor tissue, and trigger autoimmune diseases. Tremendous efforts have been devoted to elucidate the generation, evolution, and functions of autoantibodies, as well as target their autoantigens. However, reports of these countless previously identified autoantigens are relatively dispersed in the literature, so that's why they constructed a autoantigen atlas database, text mining and manual curation, extracted the 45,830 uh, autoantigen related abstracts and sentences of either autoantigen or autoantibody, lexical variants refined. Finally, they identified 1126 genes as human autoantigens and 1071 related human diseases, constructed the human autoantigen database or the autoantigen atlas database 1.0. This database provides a human conveniently browse, retrieve, and download the human as well as their associated diseases. Okay, so this is the link. Might be this is the reason because it might have got updated the versions. They are saying about this one only. Let me check. Just give me a minute. I'm not sure why it's link is not working. Might be they are updating it or something. I'm not getting the other versions as well. Sorry. So this is the website. Sorry for making you wait. I'll just put it in the chat. Auto antigen atlas. right? So might be it was giving some error. So this is the interface which looks like, right? Now if you can write breast carcinoma and search it. So these are some of the results. So how many abstracts and how many things have been. So this is what it is happening if I just in the auto antigen atlas. So auto antigen atlas is working in this principle. Yeah, so auto antigen atlas is what it is doing. It is fetching out information through literature mining and data curation. So what it is that, or what it is uh, particularly known as, you can tell. So it is searching the manuscripts. So for example, you are having one uprot array. You are working on certain disease, like for example lung cancer. You had the serum samples, and what you did after you are having the serum samples, you are running the samples over uprot arrays. So once you are done with your samples and have run it through uprot arrays, so then what it will be there? So then you are getting screening the auto antibodies. So screening the auto antibodies, publishing in the manuscript, right? So what you did, you had the disease, you had the serum samples of the patient and healthy controls. Patients. So you have the patients and the healthy controls. So what you are doing, you are taking that particular samples, running the samples of uprot arrays. So what is uprot? Having normal proteins, okay, which is having across so you have the normal proteins across. So you are screening the auto antibodies and publishing. Now the gene or protein product, a protein or gene product, rather it should be correct. Protein or gene product. The protein or your gene product which you are taking it, so that which you are publishing in the manuscript or reporting in the manuscript, autoantibodies against. So what autoantigen atlas is doing? And if you're the reported manuscript, so it will be, and it will give us code on the basis, and it will score on the basis of the number of times it has been published, disease associated with it, etc., etc. In this way, it is going to curate and it is going to take it. Okay, so you are getting that those particular things, and it is getting the scope. So in the Advantageon Atlas, if you go and search what it is saying, AMPH, then you have the disease, and you have expression level and your scores, and you can download it as well. So let us try with TP53 and search. So TP53 is associated with as an autoantigen in this many diseases with this many PubMed abstracts, full text papers, the data sets, and what are the relatively scores. If you take a disease, for example, colon adenocarcinoma, so it is CALM3 via CAM5 COTL1 which have been reported. Now if I say for pituitary adenomas, you can ask for gliomas. Okay, so these are, so you can scores, full text papers, data sets, and you can come up with the scores. So GAD1, that is glutamate decarboxylase 1, Antibodypedia, GAD1 antibodies. So you can go and do your further research on this particular things. So this is the one thing that is there, which you can take it forward for your particular understanding. Okay. So is there any question? No, right? Now I am hopeful that you have got an idea about why antibody arrays can be helpful. and these are also uh, an excellent way to say about what I can say is that to say of the particular disease, which is your uh, to say of a particular disease about the autoantigens, and it is also like the with the help of these arrays only an useful resource for the community, like the autoantigen atlas has been provided and has been added, right? Okay, and also if you want to look at this paper, so you can also go through this paper, which I had given one example, but nice one. So here what we say that about the human genome, the types of protein microarrays, which we have discussed a lot while we had discussed it in the previous phases, like the analytical protein microarrays, functional protein microarrays, and the reverse phase protein microarrays. Then autoantibody profiling, this figure is already we have shown to you all. These are some of the autoantigens or autoantibodies that have been identified from ovarian cancer, glioma, non-small cell lung cancer, gastric cancer, malignant mesothelioma, prostate cancer, autoimmune hepatitis, osteoarthritis, malaria, type 1 diabetes, multiple sclerosis, rheumatoid arthritis, Parkinson's disease, or all those autoimmune diseases, cancer studies. So not only the autoimmune diseases, but these autoantigens or autoantibodies also plays a huge role in cancer research and cancer-based studies, where you can get the markers for early detection, and if you can get or identify certain antibodies, which is actually, if you see or if you find that it's actually getting to be understood and it's getting to be analyzed and it is getting to be in uh, that particular antibodies, if I say that it is going to be coming up in early detection, in that when the disease is just starting or just expression of the disease or just when the disease is progressing, as a diagnostic marker or a prognostic marker, if you can identify certain autoantibodies and characterize them and through this kind of technologies of high throughput arrays, then definitely you are on a very good age of combating or preventing for that particular disease. So this is a huge chance of that particular things to be worked upon uh, against, right? Then we have in the PTM studies, and this also I could show you that how this particular resource useful for the communities like the autoantigen atlas could be identified and could be given there. Okay, so these are the different applications which you can easily go through it. So this is nice review article which has been published and which can lead you to the understanding and identification of all the particular things, right? So any question till here? No, right? Great. So I have given you the resource autoantigen atlas, different types of arrays, why autoantibody arrays can work, and the reverse phase protein arrays and the protein arrays which we have discussed quite a bit of time previously as well. So you have the functional protein arrays and the reverse phase protein arrays, the tissue lysate, where the you have the lysate getting uh, you are yes. So you have the tissue sample and lysing, and then you have the proteins and the antibodies further getting into the picture. So this we have already discussed. and I don't think again we need to discuss it. Now what we will be looking, so the experimental workflow of microarray, so it depends a lot of steps. So I'll just give you a glimpse from a video from a recently conducted workshop where it has been shown in great detail how the work of a microarray from a two-prod array chip is done, so that you can get an idea, right?
What the protocol sheet.
See, so this is microarrays. So you can see the glass slides. I'll start up once again.
And then we equilibrate. Equilibration is
Yeah.
After you have not from the already not to start. And what are you from? We all need the because we want to minimize any non-stringent line. We want to make the binding condition very stringent. So we bring in lots of diversity. That's what we usually get here. Schedulers. Created. We usually drop in buffer here because that was what development asked us to use. Primarily because in the draws carry steps back, and they had observed that droplet prevents or rather does not allow the signal when she prevents. If you are thinking of no microarray, again you standard. What your blind microarray design is, and if you're working with your own lab, you can do standardize their labs. We standardize it for our chips. It might not work for your chips. Usually a chip comes with a manual that these are the buffers you need, but whatever the buffer, primarily we start with blocking. You're also making some scaling solutions, and these solutions what the protocol sheet. So what we do is we first allow the people in the site. Remember these sites known as peptides. These are right and they travel long distance.
So you can do with protein as well as the peptides as well in the printed on that.
First step of working with this specific kind of microarray is allowing it to come down to temperature, don't allow any condensation, and then we equilibrate. Equilibration is align the peptides, these price peptides as well, because we want to mimic exact natural condition. Do we mimic it 100%? No, there are limitations, right? So these peptides we try to match as much as possible with natural conditions, but you say with 100% we have no there any limitations, any models. Next, what we do is we start the block. So I think we can have these time states one here. If you're see how we start with the blocking. If you don't come random, I will take one question. Maybe those are standing there.
Yeah, so you can see the exact setup.
So how the data back to us. Okay. So remember, once you put this slide, okay. I think this is okay. So once you slide, okay. Okay, so there you go. Standing, I think, randomize from the back. the one in the middle can take the 10,000. Fast function, don't pay. Right. So can I have those? Yeah, there are two things. One thing I know. Krishna. What we do is first open up the tree. Now we will put you can put these three sides. One one side. These two places. They use a similar glass slide, microscope slide. So we don't want this compound to want on. In this case, once you set this up, you are setting up on the next 24 minutes, depending on the length of your experiment. So that in the time frame, you have to print all these three slots. These slots, 3 seconds. That have checked. So I think you want to learn that. Very easy on extra on. So these sites are but they are able to see so under camera for the camera so people if you come very close to it you will see something is printed on it. It's not exactly transparent. Something is there, but what is there that we have to have very small microscope. What we do is put things like this is the bigger model. I mean, for that, what's the size? One. We have inside. So we can have a closer. So we don't want to scratch the surface. Right now, the hard time scratch the surface. Okay, this small. It's very bad fast process. Only the regular traditional process. Try to reduce them just so that it fits. Of course, the peptide rich surface will be formed. That's why we have these equal to when we know where this kind of see the like one side we have to be able that other side of those having on the side. So peptide rich surface will be formed. These are all custom made. The different make their own configuration rate. These are custom made for chips. One way of making money is just like 25 also has their own. Everyone, where these last time millimeters this smaller or millimeter longer than the traditional last time would be 1 meter long. But that way, they're asking you to buy all this. Again, they will buy their information tree dimensions. Dimension is also a dimension. This is not science. Maybe but I think what you would see is the peptide written up, these up, all these are sets up, one part, silicon gasket, the gasket. sides. One is like one is more tiny, one is less shallow. Now we try to align it to both. Now what we see is a particular area is getting inundated. Another part of the site is being spread. Also, if I put patient works here such a here, patient here, there won't be any reason. Now, the put this one, and then one thing that starts storing. The idea of the storing is to the time. This, 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 that, particular. The internet will operate your 60 pounds is like because we were traveling. This is more of business. They're more traveling life. Space and space is there. So the maximum is to be on the stable side. We use good. The reason we not use much more volumes, why? Because in the next step, you would remember, that we need this overnight in the whole room. Take a very maximum volume for gravity. But we don't want to put less than 2 minutes. The reason is we leave it at 60 for almost 60 hours overnight. And sometimes the air is dry. We don't want the slides to dry. This is one big rule working with micro, especially the pen line. The first step of equalization, when you're wetting the slide, is the last step. There are very few steps where you allow the slides to dry. The slides dry, the reaction can be slides the logic and the figure. Somebody else can dry. Actually, can not take that now.
We have only small here. If anyone has small,
okay, so once it is done, we have the right. Okay. This is the first step that you need to do. Bring out the slide out of your storage. Bring it into the fridge. Allow it. Bring it to 50 and 90 Celsius. Then you want to avoid too much of moisture. Keep it at 25 degrees Celsius. And then you flash before Then what do you do? We have a cover. Right? Cover. Become a boss. Many of you would be doing this kind of integration in right? You put a take a small glass rod, put one slide, allow some water, then you cover it with another. But we cannot do this with this slide, only do very specific. The volume is required, what proper is that we require. That's required. So, find the line over the chips. The first step would be block. How long does the blocking take? It ideas. You can go back to the video. You can go back to the I would ask to rather go through all the entire video. I think this is the washing buffer, which is also used as an equilibration buffer. These buffers are the constant. So again, the type of slides you're using a neutron slide, which is a protein macro array, not a peptide macro array. Your buffers will change. For peptide macro array, if you are using this is a neutron chip. So the chip will contain positive. This is also a macro array chip. Calculate the camera. So is it the chip that we were seeing? In fact, you have very sharp vision. You can see it's mirrored, right? Acts like a mirror. Different. So every chip manufacturer will have its own specific protocol. But all these protocols will basically follow a modified protocol. Right? So some of the things you can spread this. Try to observe the various patterns that you can see. Try to reflect the data on the surface. You can actually see in few of the slides, you can actually see the peptides being printed upon blocks and these boundary blocks. Yeah, just be there. Don't break it. That's the only difference. These one chips, these are also used. So we are getting it clearly before completing the any road that does not have any person these are new processes. These are the ones that are the entire human proteome is printed on. These are the chips that you want to use, that you want to understand what the antibody. Right? Proteins are already printed. The serum that is antibody is a human protein, most likely is a normal antibody. That can be used as a diagnostic tool. It can be used to prognostic. Right? I hope most of you would understand diagnostic and prognostic. Diagnosis, when you use something prognosis, what is the fate, what will happen next? Do that. You can come back. That's history prognosis, but that's what you will do. Especially in cancer, things like cancer. It's extremely important to know how long it was do you focus? How many people this sort of thing? But too late, when it's 25, 50, 90. So decisions that patients are taking, science, right? So these are the chips. Shashna, I think we start with the big. You have seen the approach to Pablo. And I have an approach here. But as with most proteomics work, you need to carry your icebox around with you because you don't want to denature your sample. The peptide, it might be traveling from Germany or from Frankfurt. You can take a flight under dry ice. But your samples, they need to be very robust. Again, especially if you're trying to work with antibodies, don't freeze your sample. Sometimes we could get overboard, we freeze our sample. And the centimeters they say, कभी खराब नहीं होगा. No, if it is an antibody, it's a pretty large molecule. 2, 3 freeze from cycles, all your antibodies are gone and gone forever. The sample might not be. So you need to remember when you design your experiment, what kind of experiments are you planning? Right? Are you trying to find an antibody for freeze? You can just put it below freeze. Right? So these are the things that we need to, and these are very specific for each experiment. We've seen these incubation tray assemblies, and right now we are using solutions. We are doing in this room. Ideally, we would do it in a lab now. If you don't want to pick up that or any foreign contaminant falling on your chip, and we are using right, almost like an iPhone. So we will always use it under clean conditions, aseptic conditions. Right now, these are used chips. You can use it any way you want to because we extracted the data. Sometimes we might not have 3 chips to work with. That time we use blank chips. If you see, we just put a blank chip that does not have any diagrams associated with it. These are balances. Physics, you don't want the rubber gasket to leak. If it leaks, we want to prevent the leakage. इसकी बंद कर दो
26 को 21 कर रहा हूँ 21 कर रहा हूँ
So this is the basic protocol, right? What we do next, we use our poppers. Once we have done this, one of you can try and put poppers again. This is not leaking. It should not fail. Also, one fact that we learned while doing our experiments, it's never wise to ignore these 3 things. Why? Because sometimes there is the slide of skill. So you need to test your skills, what you can do it, and keep the balance. But if it is important, we keep it for operating. If there is a leakage, then you will see there is no approach. So all the steps that you do after this, you are not going to see it, cover it, and be in a lab room, or room, or cheap. After 16 hours, you might feel that whatever damage has been done, you cannot always observe. The entire night, there is some change. And even if there is leakage, there is nothing to do. Okay? Because we cannot have to take out the size. Because then the night, get dried out. So whatever before you put a beginning experiment, these experiments are big and what is happening because once you start, they won't stop till you reach the limit. There is no pause. It begins between and at the very end. No outside nothing. So So I will take a video to be work with us. She will tell you what we do. The first part is blocking. Block the after block lab, we dilute the serum samples. Let me speak about that. So most of the serums have a lot of antibodies. We do not directly give it. We dilute. We don't want too much of antibody because too much antibody can lead to distribution buffer, which is basically the same buffer, and one magnetic distribution. Both of them we can work with very low volume. We don't need to draw blood from the patient. We can work with leftover blood. All the work that we did during the COVID or COVID-19, we were basically leftover blood. Only technology have also mastered this art of working with pioneers and making it happen. Right? This we did almost 2 to 3 times we keep having to get higher from the top. Plasma techniques, antibodies, DNA, cells. Right? So here we go.
We started with this is now the slide. We begin with the buffer. So the buffer will have whatever was projected as less and some percentage and we have similar impact. And that is followed by blocking. Blocking buffer has mostly speed, but including PSA and some protein. So what I do is for the purpose of demonstration, I will have the net content step I will add the staging buffer, the watching buffer. Maybe go to another movie and the end of the whole and not that we want to use that there will not be whether the basket has been mounted properly and the screws are tightly and then there is no when we are satisfied with this, we can keep and add the bottom of the other side here. If not, we might have to just set up the relationship.
For 15 minutes simulation, if I can put in the video, because that will give you an entire process. The 2 days, the next 49 minutes.
One side because that would not be properly or the two was tightened just very tightly. So because of too much pressure the side and
Watch the video.
so we just put it on and off. That's the process.
So first we will equilibrate it for 15 minutes, then take the buffers, right? Then we will add change of one of the buffer, and this buffer will be again put for a 30-minute increment. This is the blocking buffer. Then we start the sampling phase. The second is buffer. So here we use a 1:200 dilution of the patient plasma. Again, we dilute it, allow it to. These are not frozen, right? Yes, in fact, 4 days session where we would bring when we want to structure, from what we bring to what we want to study. these slides, these steps are now done with the data. The reason is some of them done technically as important data. So then it being labeled. Now we have, and we don't want to see. And the type of secondary antibodies that you want to solve depends on the type of secondary. Right? For the very active, very active, if you're instrument and three different you can use different antibodies. There are very common things that are there you can use three signals that we do all three different antibodies. One is like one group can antibodies A, F, IgG, or whatever IgG you want. Right? I think you can play out the rest of the video. First time we want to speak of like that. Okay, so what did we do next? After the entire stage was done, we take like now. Then all that we did in a last class, this is the topic. The last round. Okay. Here we put all the sides together and we want to again in the second way antibodies will not off. Unconjugated fluorophores. We don't want to see the unconjugated. We want to see only those from nitrogen. Water vapors have a lot of salt. What happens when salt is drying up? The precipitate, right? Now it will be doing the scanning, and that precipitate will come up and take wash. So we wash off the vapor, washing vapor, taking vapor, which right now is automatic filters, and then what we use a pressurized stream of nitrogen. Basically, we have an attachment that comes directly from the nitrogen cylinder at a very high pressure. If you're either using the hospital, you will see a hand dryer. What does the hand dryer do? It goes air at a very high speed. Serves at a very high speed the water moves off the surface. It dries by pushing the water off the surface of the valve. Similarly, when using very high pressure stream of nitrogen, all those remaining droplets in the micro droplets, we are removing the vapor this time. They don't allow it to air dry as such. The pressurized stream of nitrogen, so that stream pressure, the nitrogen, the water droplets, along with this, everything moves it. We want the very inside, we only want to see the binding. Just with the rest of the video and then. So this is what we can see. This is the microarray scanner. And what we do is put the slide inside, and then with the help of the software, you're seeing is this is actually a recording of a live scan that we did for the video.
That's it.
So we can actually, our teammates are there over the lab for this. We can give a direct demo of that. Amrita, are you there? Can you start the camera?
Yes, sir. You're in the lab and I'm starting the live demonstration from you.
Yeah.
Yes. So I think you have got a good overview of the experimental workflow that using the microarrays, how you can do that. So one thing that uh, we have talked about mostly is the protein microarrays and the proteins, but this kind of peptides, so with the peptides that are made of, which constitutes the whole protein, can also be printed to get you different. If you have overlapping peptides that gets printed upon the arrays and the chips, you will also get more different information, among which one case study what you can consider of, or what you can think of is the recent report which had said that the immune response profile of the COVID-19 from the variant specific epitopes from this kind of peptide microarrays, of which you have seen that how a particular experiment is designed and experiment is done. And now we'll also look at how the data analysis and the extraction procedure is done, and finally the data analysis is done. Before that, we'll move into the manuscript to look how a particular thing is there. Yeah, so let us look into the paper, right? So this is the abstract where you can see that the serological diagnostic aids. So we, the authors have studied the humoral human response of SARS-CoV-2 infection to map the immunogenic regions in the SARS-CoV-2 proteome. So this, which are the, so you can think of an application in a such a way that which particular regions are immunogenic, so that you can target those, you can vaccinate, you can prepare the vaccines against those, so that it will give you an overview and you will give an overall idea of you to take it forward. So this is quite an important in a way to have for your better understanding of the different regions. So the microarray technologies, high throughput technology are the platforms not only at the protein level, at the peptide level, or the single amino acid resolution, can also provide you very information, of which we are seeing one classical case study. So the resolution using the microarray has now that's 4932 overlapping peptides printed in duplicates entire SARS-CoV-2 proteome, and they found out different immunogenic peptides against IgA, IgG epitopes of these six would discriminate between disease severity. So how that has been done? So the materials and methods, methodology or the experimentation procedure of which you have seen the video is the way that has been done using equilibration, then washing, then primary incubation, secondary incubation, and then finally scanning, of which we again are one procedure in this particular week. You have also seen in the lectures that how to take from a protein microarray using a Huprot arrays the technology, the data extraction of the data. Also, we would see in how in the peptide microarray cases as well the data is generated and data can be taken. So just if we go to the brief of the peptide microarray. So what is a peptide microarray? So apart from the protein microarrays, now the peptide microarrays are also in play for a long period of time. So the epitope level antibody responses were deciphered. The it was the entire proteome of SARS-CoV-2 isolate. The protein sequences were translated into 15 amino acid long peptides with an overlap of the 13 amino acids. So 1 to 15, then from uh, it will be starting from 3 to 17. So in this way, 13 long peptides are overlapped. Individual was thought and then diluted. So all these steps you have seen in great detail, that how it is to be done and what is the role of which buffer, of what is the role of each buffer for this particular experiment, and each and every step needs to be performed in a very great cautious way, because one thing is that not only the experimental design and the experiment is quite tricky, as well as the chips are quite expensive in nature. So for which you need to provide your 100% attention to get it done. So now the for the experiment, and then equilibrated using the staining buffer for 15 minutes. So you have gone all those through all those steps which are required. The slides were then incubated and then done on orbital shaker at 140 rpm. Slides were washed thrice using washing buffer. So washing buffer with that composition and that thing has also been shown to you, and how it is done, that is also shown to you. Then was aspirated entry after each wash using a micropipette. The slides were then incubated with a mixture of Sci5 conjugated rapid anti-human IgA and Sci3 conjugated donkey anti-human IgG, and then it was done in a following dilution, and then it was again washed and the incubation was tray then assembled disassembled. So you have seen that how the particular tray is also being assembled and the screws are tight. So you need to be very careful, because with that uh, abnormal or absurd amount of pressure, you would break the slides, which are quite expensive in nature. So you need to be quite a bit of careful in that manner. Okay, so that needs to be carefully done. Then slides were processed to acquire signals. Then the data acquisition, images were acquired, and the fluorescence and different data analysis technique. So here what we can see that the different samples, heat treatment, washing, incubation with secondary antibody, washing, and then particularly scanning the slides. And then background correction and the data analysis is the next part, which we would again see. And then the results. So just see that what are the, so in this expression of the IgG, IgA, what they have told, the heat maps showing major immunogenic regions identified in the SARS-CoV-2 whole proteome microarray constitutes polyprotein encoding 16 non-structural proteins and structural proteins and the accessory proteins, and then finally some severity based response. You can see that the what is the differential expression of those particular peptides which are showing to have a good immunogenic responses are being done. Okay, so the mutant and the this wild type and the mutants, different analysis. So now we would be looking into that. So I'll just, so how the data extraction procedure is to be done. So you can see this is in a real-time manner, and how the data extraction procedure takes place. Okay, so these are the some of the spots which you need to align. So this is aligning to extract the fluorescence intensity out of each feature, out of each proteins, or out of each peptide. So here in this kind of arrays where you have the proteins got spotted and you have placed the serum and you are getting the antibodies, then you are getting the fluorescence signal intensities, of which you need to extract the feature out, extract the intensities out. So I think this intensity has been also discussed thoroughly in this week's lecture, and this is the peptide microarray. So after you have got the features out, you have aligned the grid, and for each spot you have extracted the intensity out, then you are subtracting it with the background noise and then normalization, which has been thoroughly discussed in this week's lecture. You can go through it, and this is how, in real manner, one can uh, one can scan and extract and try to align the grids and take it forward. You can see the extracting the features, and this you are seeing in the lecture, and this is how in real time manner you can get it. Yes, so hopefully you have seen it. Otherwise, I can do that once again. So before that, let's go into. So now we will see the data analysis steps.
Meanwhile, Western blast, transmission electron microscope. How we have started to learn this time microscope. This part as well.
Yeah.
And also back. So those who have installed, they can do that. Otherwise, lunchtime I can use some data I can cover if you use. Yeah. So microarray, the high throughput.
Now you can look at that after the extraction, data extraction, how you can take it forward for the analysis, which was again shown in great detail in the recently conducted workshop.
Coming to what you slide might be this microarrays followed by microarray image, how this looks like. And already in the demonstration, you might have read that I have given a demonstration of the lab when I turned on the scanner. So there you could see that when we are switching on the system, then finally after the scanning the very end of the system. So then microarray data extraction, how to do the data extraction, that you can see I will the uses from the PPT, microarray data analysis, and finally hands-on now. Yeah, so this I think this is also your completely available already uploaded given some about this yes already talked about it. So now so this is kind of the ideal proteomes which is painted on the chip onto the chip on the this website are printed in 40 years back. So 15 amino acids, 50 more amino acids. So that I will also show you. Then we have analyzed two samples that severe and non-severe. And finally, after interaction with the biological acids, I usually engage in the design part for this purpose. But whatever is micro, protein micro, this is the GP scanner that you already got the idea from lab. Then after we are getting this images, this is the green channel and this is red channel. After you inform me about this is for this IIT investigation, this is for IIT. This software is important. This is the GP Pro we are using in our lab. We are using scanner, this website analyzer as well as graphics. This software is very used. But initially, this part generated this image generated in GPX Pro. Followed by we are taking for this mappings and website analyzer and GPX Pro 7 for data extraction. Then finally, after getting the file that is called comfortable file, that is called GPX result file, that is GPR. That file will be taken into R or other than. Now, this is the image. I think you are also got this idea when I already scanned this might have So I'm just keeping this one. So this time control algorithm synthetic already observes completely managed view because these are the specific algorithm that is required to excite this tags. I think here also mentioned here in the category antibody and conjugated tags. So there are the lasers actually excites and you require a signal that is for the process. By the way, actually I am developing on this electrical area. I am developing this kind of instrument that is very brilliant in my research. And parallelly I am working in this microdata analysis. So now coming to the gap file, this is very important. The gap file is required to extract the data. This is simply a CFD file. This CFD file will just take out. This will be nothing but the LPD. How these peptides are related? We are working with this 10,000 peptides that we need to inform the specific company who are delivering this kind of thing. So they actually provide us this kind of particular CSV file from that is the we contain with the specific peptides. But there are not only these peptides are there, they are giving lot of other parameters like the coordinates where these specific features are associated. That information will also get from this software. But this is also very research related problem. Actually, researchers also looking into this to extract the data without using this kind of features. This is also even costly problem. I'm also looking into this part as well, specifically another project. So this is also completely computational. Now you can find this kind of this is actually what gene tags for the DNA. But if we can get the idea for DNA and gap-file, then finally we can proceed for protein, peptide. It will be like a context. Just idea is bad. How we can grasp that. Now we can find that A is equal to X or G equal to Y or G. We just change it. And they will be that we are also create our own gap. Whether we are not dependent on any company, we can create our own gap in lab. And finally, placing on the either damage or the extend the. Now, if this file is ready, this file you able to manage properly. Then there is some lagging here. So now, after placing the gap file properly, this is the placement of the gap. So this is kind of gap file used plan. It needs to be placed properly on the image. Why? Because if you do not place it properly, the grid on specifically this box are for the feature and technically the grids are for feature indicator. So if the feature indicator is not placed properly on the specific spots, it may lose the image. That will be bad. So this is the reason this actually requires a lot of time. No automation, nothing. We need to scan in front of the system, place the gap properly, and extract the data. This you can find for IIT and IIT. Because I am focusing right now today for the peptide microdata. Now, if this gap file is ready for let's say GPX Pro or Navics or whatever the data extraction software is, then this part is ready. Now we need to just go to the system and it will give enough time to show that when I am demonstrating on the system. Then in this platform itself, in the software itself, there is a specific tab that is mentioned as data scan. If we just place on the data scan, click on the data scan, data will come. So that data is specifically called as GPX design file or GPR. This GPR file looks like this. There's some lags. Yeah, so this is the GPR file. So now you can see a lot of proteins, this peptides, this idea. Actually, in the last one of you get this peptides. So this XY coordinates you are getting. This information is quite crucial. This fluorescence intensity and background. Because the image you can recollect, there is spots. Now the spots you can see some spots are very dominating, it's giving you a signal. But still it is also having some background there. If we have some background, it happens. Sometimes the suppression matters from this point of the background. Now, how researchers begin the form? They started with good minus polynomial density minus background. But lot of skills are associated for data analysis. So what happens? Let's say if we are doing this f minus b, sometimes we are losing some kind of intensity because data state is completely mathematics. Data state you may go for the logarithmic conversion. So let's say fluorescence intensity compared to the this intensity background data science point of view, we need to take it further, not at all matter. So, what I found, just looking to this specific, this is actually an amino acid sequence. So, this is actually a peptide. So, here you can find some specific location. Then you give this. So, I try to find that what is the next one. So, they have found, so they have actually taken their data. This is from the pre-process data into JETS code. So, that is normalization study. So, for peptide, this JETS code technique is very useful. So, which I found, so they are getting this converting the data set or standardizing the data set into JETS code. Now, after getting the data set, they have taken a threshold of 3. Why these 3 have been taken? Now, I will start to be different reasons why they have taken 3, but it is statistical concept. If you know regarding this Gaussian distribution like this, so if sigma is to be this, x dash is and delta is this, so sigma over sigma, this way you can write it. So the data which is coming from the tail region will be taken as outlier. So that means if this is an outlier like this, so definitely it is having some significance. So that means the intensity, whatever it is getting much important. So this is called 3 sigma. If it is sigma, it is 2 sigma, so it is 3 sigma. So why we are taking J becomes 3? Because 3 sigma is there. So now what I realize, after converting the data set into Z scope, mu becomes 0, standard deviation becomes 1. You can also look into that. STD becomes 1. Now look into that. So mu plus this is mu. So mu plus 3 sigma. Beyond that, that will be taken out. So we don't have to replace with 0. Sigma will replace with 1. So 3. That is perfect. So we can go ahead. This is my code. So I don't have the equation peptide because they are interested for that. I will take from the data set. This is 3. I will take it forward. If for patient, sorry, this is basically number of patients are there. And anyone is the peptide 1. So anyone if getting more than 3, greater than 3, that will be taken as simply that will be taken as immunogenic peptide. Now let me consider it as inverse matrix. So this is let's say 1. So P1 or P2 like this. Why I'm telling that? I will tell you. So this is the one. Finally, this they also ask me this another question of we want to find the discriminatory peptide. So what is the discriminatory peptide? For discriminatory peptide, we need to find to analyze the data for severe to non-severe. So some patients we have also taken severe, some patients are non-severe. So that group also to become compared, and we need to find that which is significant. So that significant for when we taking in age for forward, we need to know the p value significance. So what p value? The threshold should be usually for the biological data, for the protein you said of you take or micro data. This threshold should be usually maintain p less than 0.05. That usually take. If this is usually case, p less than 0.05, I need to find out from the analysis as well. How many peptides, when I am considering this severe and non-severe, how many peptides I am getting? This satisfy this criteria. But again, different normalization strategies, different statistical analysis are there. Again, you are literature reading the books. This is called the non-parametric test because we are taking the Z score forward. This is the reason student teachers don't care. You can't take this analysis to student teachers. So another what are the other strategies are available when you are dealing this non-parametric test? That is either magnitude test or Wilcoxon sign that basically the same, which is to compare it when we just mentioned either paired or unpaired. I'm showing. So when I'm showing the arm. So there we need to take that one how many peptides are getting a significant. That means my biological colleague who are from biology, they are asking this question. If I give this the number, they are satisfied. So this is the way project would be taken. Let's say I can't be like maybe I'm not much aware of the biology, but the data science part is also very, very important. And computational intelligence is dominating in these days because of the time being. So how we can minimize the effort, minimize the time. So next part is this that the Z-score values, and finally taking point. Now, I already mentioned about the specifically this part of this outlier part, but you can see this part. This is I already told that steps like this background current tape followed by this averaging, then logarithmic conversion. So you can get this is the commands that have been used for the specific island. So this is the XD mean we are getting. Then logarithm we are converting into log. This you can see this log here. This is the data frame that is converted into logarithm. And finally this Z-score here. So this Z-score is nothing but this J log here minus ZSP divided by ZSN. Whatever I mentioned that actually there it is taking as the data frame. Now after that, this is the mean max scaling, because sometimes this Z-score is also taking it as a standard edition, but this is outlier dependent, this specific Z-score. So that's why we have taken Z-score for the parameters. Now again, this specific algorithm is very much important, because should we go ahead with the specific normal L minus B strategies, or we will create something? Because I have also worked with the protein micros, because I have built 2 more projects in protein micros as well before. This is for peptide. So this part was quite interesting for this family evaluation. This was really we have implemented in the peptide micros or our partner. Finally, this thing I already mentioned, this specific algorithm. Now, these are 2 data visualization strategies. That is, you need to know about the interaction with the different. This is called QC, followed by the parallel plots. But this ggplot I also mentioned. So ggplot is completely giving this dominating this and it is available now. So finally, this kind of plots you can generate, and you can see actually eliminate some outliers from this analysis. So this is the section of code, and you may also get into the code part where I actually what I said. So this is the peptide which we found. Now, again, this is called the EPO. I think also mentioned about EPO. So now you can understand more about EPO. Contiguous peptide. That means immunogenic peptide was my first question. I wanted to give them last. Next question they raised about the immunogenic EPO. That means again a code is required. That means you can see this is a contiguous we are getting. This like so contiguous peptides we are getting. And this is the reason you are saying it as a contiguous peptides or EPO in immunogenic function. So immunogenic EPO. This is also computation compared. Now finally this part is the statistical part, and these box plots are very important because finally you can see that how many stars you are getting. The star signifies this range. This is important at this point of whenever you are analyzing data. This you need to give this star marks. So 1 star means they will able to understand who will review your work on paper. So this p-value comes between this range in this range. And this one second 2 star is like that, and 3 star means below 0.01. So this way we have taken this project, and next part is another important. Let me just give you some, because whatever I told you about this unpaired sample, 2 groups might be the 3 groups. This is an ongoing project. We are now working in this project. It's a new project. We are working with the longitudinal sample. Almost 50 samples we are taking this time. So there you can find something very interesting. This is very interesting statistical analysis. Pushkar-Wallis test. So just like ANOVA, you might heard about ANOVA. ANOVA is related to working with the parametric test, and the Z-score is non-parametric. For Z-score, if you are taking your data for the Z-score scaling, the standardized form, then you need to, if you are multiple groups, analysis is mandatory. Then this Pushkar-Wallis test can give you the idea of which peptide is significant amongst this. And intergroup also, that means also that is crucial to understand. This is a theoretical perspective. Now, all that if you want, that can be given as the Pushkar-Wallis, but individual if you want, that you can get from dance test. This is also we are taking for our project. So now this is our just ongoing one. I wanted to give you some more clips of that. How which Z-score that we are taking. And see in the parallel you can see 2 significant. This particular peptide data, you can find this peptide data, dot data, peptide underscore data that already we have created on data frame for that. That has been taken. This is the only thing. Yes. So that file, whenever you are storing that file, that location has to be given. That's why I say the first line is equal. In my case, okay, let me demonstrate that. Right. In the folder itself, how many files you can find? 2 files. One Excel. Yes. Where you have stored that Excel? That location is equal. In my case, you just look into that. What I will do, everyone just check that. I will just taking my cursor here and click here. Got it? Control C. That I will paste here. Okay, but in the next line, when I fix this ZWD, I need to change this specific slash. This is the trick. Just find it out. Then right now. Hello. Yes. So now I will go to this. There is a question that how this peptide. Yes, sir. That will be written. However, that is there in the what is ZWD in bracket in the first bracket. Yes. Finish now. So can I just first show you the code, then I can come and give it out. So
Just one of the class one I will repeat the
yes. So in ZWD, here you just okay. Again, I'm repeating this part. I am going to the folder. Okay. In this folder, whatever you are storing this, keeping this specific 2 files, R41 file, and next one is this CSV file. Here, just you can see the my cursor. I'm going there. Just control C. Okay. Minimizing this screen. Then here in this ZWD, double quote is mandatory. In this ZWD, you need to paste that, but you need to change this slash. This is the requirement. Then only it will be. Otherwise, you won't. Anybody just do that? It is possible. You can do that within 2 minutes or 1 minute. Okay. So when I copied that, okay, let me again just to give you one. So whatever I have copied from that folder, control P and V, because I have taken control C. But this writing no need to be run. So I am giving a this dash. My system is there. Yeah, but so I need to run this line. So now again, I just copied and pasted in the center inside the double quote and changing this slash symbol. This slash symbol in this rotation I am providing to this. That's normal change. If you do that, then this very library I already mentioned that might has to be installed. Otherwise, it will take a lot of time. you already installed this file, then what I in a during time record. Analyzing using all these softwares. Uh, no, so that's why I'm saying I'm coming here. So just let me go to this. Because I think naturally progress. There is record. Everybody got this. That's what I'm saying. So most of them are missing. So I thought, sir, I'm doing it for this is what I want to see. Yes. So yeah, tasks. What is the first EW?
So basically, what I can say here is that you can do this try this kind of analysis.
Underscore. Already it is coming. You can see the database itself ratings on the system.
So basically, you can try to do. So here, what we have uh, told is that, or said that what you can definitely do and try out is that you have seen the workflow. So we have today not only discussed about the proteins, protein microarrays, different types, although we have also looked at very high amount of source of auto-antigen atlas. The second is that in not only the protein microarrays, but this kind of high throughput system can be used in peptide microarrays or the peptides for different applications like immunogenic epitopes and others. Here we have seen the complete workflow, how it is to be done, washed, and then incubation, and then finally the data extraction. So we have seen that how the data is extracted, and then the data is analyzed and further final analysis to give us the results. So hopefully no question, and also the quality, array quality and the chip is very much important. So R value having the reproducibility in the duplicates, the R value or the rank correlations values of the square manipulation should be more than 0.95 or 96, and that should be important. Okay, hopefully you have no questions, then we can meet on the next week, next Tuesday. Thank you.